Assalamu alaikum, everyone. My name is Maryam Altayeb, and I'm a final year medical student this year. And so I'm going to be teaching you guys about variations in inheritance. So Hiba Jamur gave you the first part of this lecture, which talked more about the patterns of inheritance. And inshallah, I'm going to try to make this as simple as possible now that you guys understand all the other basics. And feel free to always let me know if you have any questions in the chat. I'm happy to stop and happy to repeat anything you guys need at any point, all right? We may be accompanied by my cat at some point in this. Let me just, there you go. All right. Because she's sleeping, but if you guys hear any meowing, that's why she likes to join in PAL sessions. Okay, so Bismillah, we're going to be talking about variations in inheritance. I know it seems like a very dense lecture with a lot of slides, but honestly, we're going to simplify it as much as possible. Let me just do one thing before we start. I don't like the animations. They take way too long. Okay. There we go. So first, we're going to talk about modes of inheritance, or essentially it's like looking at how many defects or how many problems in genes does it take for you to see the result of a disease. So what we're most used to, especially within Punnett squares, is what we call a monogenic phenotype, meaning it's only one mutated gene. Okay, and that's due to a single defect, and that's what we generally call Mendelian genetics. Okay, because Gregor Mendel is the one who established that. Okay, that's good. Okay, that's fine. And then you have something called oligogenic. So oligo means few. So defects in a few genes that lead to whatever clinical manifestation it is. And then polygenic is due to a defect in many different genes. So again, poly means many, genetic refers to genes. But the most common type is multifactorial, meaning it's not only genetic, there's genetic and environmental factors that go into it. And you can think of a disease like diabetes that's very prevalent. Of course, there's a genetic component that goes into it. You're more likely to get it if you do have, let's say, a family member that does have diabetes, but it's not only limited to that. So it's multifactorial. Also, another thing, I added as many MCQs as I could in this PowerPoint, and also the things in red are the things that were focused on in previous MCQs. So just make sure you guys hone in on those points. Okay. So this is just showing you a variation. So it looks like this could have been, let's say, autosomal dominant, because you can see, you know, circles are the females, squares are the males, that it's here, you can see that, oh, it's almost in every generation, but here it skipped a generation. So that's what lets you think that, okay, maybe there's a specific type of non-Mendelian pattern that's happening here, which is what we're going to talk about. If that was a bit confusing, don't worry, we're going to go through it as we go throughout the lecture. So we have stuff called modifier genes, meaning that monogenic traits, remember mono meaning one, genic referring to genes, these traits are not that simple, although, you know, we wish everything could be black and white, but of course, as with life, there's a lot of nuance. So what happens is there's modification of these genes that affects its expression. So what we're trying to say here is that when we look at a gene mutation, we look at a disease, these things do not work in isolation. It is not like, okay, you have a mutation, you have the disease, that's it. No, there are a lot of interplay between different genes, especially if they're sitting neighboring to each other. One gene mutation can affect the expression of another gene, and so on and so forth. And the main types of genetic modifications we're going to be talking about today are penetrance, expressivity, and pleiotropy. Okay, so these are the three that we're going to talk about, and you guys need to know every single one of them because they do come a lot in questions, and it's a generally important concept in medicine in general. So first, let's start with penetrance. Okay, so when we talk about penetrance, it's basically saying if I have, let's say, 50 individuals with a disease, 50 individuals with a specific mutation. And I'm sure they have that specific mutation, meaning they have that genotype, right? They have the gene, they have the abnormal gene in their system. However, you find that not everyone with this mutation shows that they have the disease. They might have the genotype, they have the genetic component, but they don't have the phenotype. They're not showing it clinically. And so that's what we call penetrance. There are some diseases, for example, Huntington's disease, which is autosomal dominant, has 100% penetrance, meaning that every single person with this mutation will have this disease. There are no exceptions. That's what it means to have 100% penetrance. But you have other diseases that may have 70% penetrance, meaning if you have a room full of 100 people with this mutation, 70 of them will show the disease, but those 30%, those 30 people will have the mutation, but will not show the disease. And so that's what we're trying to express here. So let's say you have 100 individuals, okay, and all of them have a specific abnormal gene, whatever it may be, okay, but only 70 out of these 100 actually have the clinical manifestations of the disease. So that tells you this has 70% penetrance. It only penetrated or got through in 70% of the population with this mutation. So just remember, when we talk about penetrance, it means they have the genotype, they have the mutation, but they're not necessarily showing it. They don't necessarily have that phenotype, okay? And so in the pedigree that we can go back to, we can say, okay, this is passed on from female to male and then also from male to female. However, maybe this patient here was marked as not having the disease because it had low penetrance, because it had a specific characteristic that, people didn't realize was there, or they specifically did not have that manifestation of disease. Let's say if I told you this disease had 70% penetrance. So again, just to recap, when we talk about penetrance, we're talking about what percentage of people with a mutation will show disease. So in this pedigree, for example, maybe this person has the mutation because they passed it on to their offspring. But just because they have the mutation does not necessarily mean that they will show those clinical manifestations of disease. Is that clear for everyone? So again, mutation does not always equal disease. That's why we have something called penetrance. Yes, no, maybe. I can repeat if you guys want me to. Okay. So that's the first component. We talked about penetrance. Now we're going to talk about another concept called expressivity. So what does it mean to have expressivity? So it's how much of this disease is expressed. And so a very widely used example is something that we call neurofibromatosis type 1. Okay, so neuro meaning affecting the brain. And fibromatosis, they basically have, as you can see here, these cutaneous or skin lesions, like they're almost like very many skin tags. And so when we talk about expressivity, these two people have the neurofibromatosis mutation, but in this patient, it's very mild. All you can see is this spot on their underarm. And in this patient, as you can see, it's very severe. So when we talk about expressivity, it's where are you on the spectrum of this disease? Do you have a mild form of it? Do you have a severe form of it? So what is the extent that this genetic defect is expressed in this patient? And even with this patient that has a very mild form of disease, because it's very mild, this might be completely missed in a physical exam, and they might think, okay, this patient doesn't have neurofibromatosis, when in fact they do. It's just a very mild form of the disease. Whereas this patient, like we saw, is very severe. So it's just showing you that you can have a spectrum of clinical manifestations. Okay, so we talked about penetrance, what percentage of people with a mutation are going to show disease. We talked about expressivity, which tells you people with the same mutation can have maybe a milder form or more severe form of the disease. And then last but not least, we have pleiotropy. Okay, so when we talk about pleiotropy, it's basically talking about I have, or but whoever has this disease, okay? In some people, it might manifest in a specific organ system, and other people might express in another. For example, and the example that Dr. Zuhair gave you guys is Marfan syndrome. Some people with Marfan syndrome might have only ocular or eye manifestations. Other patients may have cardiac manifestations. Other patients may have, let's say, bone manifestation or whatnot, because it's a problem with a lot of different things that you guys don't have to know right now. But essentially what we're saying is a disease can present in several different organ systems, and it's not the same in every single patient. That's why we have a list of clinical features in medicine. It doesn't necessarily mean that the patient in front of you is going to have every single thing on the list. It's not like a checkbox that you tick off. It has a variety of different manifestations. So when we talk about pleiotropy, it's basically a term that represents that diversity, all of these different organ systems that can be affected. Okay, and again, this is the same, what do you call it? I completely forgot what they're called, but you know, the same diagram that shows you maybe this patient, again, could have had a very mild manifestation of the disease, but they didn't pick it up. And so it could be due to pleiotropy, like it affected an organ system and not another. So that's why it's important to really look into what these genetic diseases are, especially if they follow a non-Mendelian pattern. Okay. So if you guys are still with me, these are questions that are very important and that can definitely come on your exam. So if you have this case scenario here, 3 siblings present to your clinic. The first had spots on the skin of his arms and legs. The second had spots on his abdomen, and the last had spots all over his body. This could be explained by which of the following principles? Is it penetrance, imprinting, expressivity, pleiotropy, or anticipation? So
C.
Okay, thank you guys. You unmuted, which caught me off guard, but yes, it's expressivity. And so again, let's just go through what the definitions are. We said penetrance means that not everyone with the mutation will have the disease. Okay. We said expressivity is the spectrum of a specific disease manifestation, which you're right is the answer here. And pleiotropy is talking about how it can affect different organ systems. Okay, so you're right. The answer is expressivity because you can see it's a spectrum. So one has spots on one place, the other has spots on the abdomen, the last, the last had spots all over his body. But regardless, we're still talking about the cutaneous or the skin manifestations. If I brought this question and I told you one had manifestations in the eye, other one had manifestations in the abdomen, the other one had manifestations in the heart, then you can say this is pleiotropy, right? Because we're affecting different systems. Exactly. Okay. So another question, both Saeed and Saud are affected within the same autosomal dominant genetic condition. Saeed has skeletal manifestations of the disease, while Saud has skin features. This observation is called?
Pleiotropy.
Exactly. So this is what we call pleiotropy. And just keep in mind that what we're talking about with penetrance and these things, we're mostly talking about autosomal dominant conditions. Okay. So, all right. And so now let's move on to the next part. So a gene may have monoallelic or biallelic expression. And I know a lot of the names, especially in genetics, might be a bit intimidating, but just take them at base level, know what the basic parts of the word mean, and it's going to give you the explanation up front. So for example, mono means one. And when we're talking about allelic, we're talking about alleles, right? The different types of genes or the different, phenotypes that we have and genotypes. So monoallelic expression means it's expressing one allele. Biallelic means there's 2 alleles, okay, that are expressed. And so we have a very important concept in genetics called genomic imprinting. Okay. And so what we mean when we talk about imprinting is that some genes you get from your mother, right? And some genes you get from your father. And when you have something called maternal imprinting, it means that when you receive a copy of this gene from your mother, it is already inactivated. It's not functional. Okay. And some genes you receive from your father that are inactivated. So those are paternally imprinted. Okay. So maternal meaning mother and paternal meaning father. So maternal imprinting is when the allele that you receive is already inactivated. Okay, and the paternal imprinting is when the father's allele that you receive is inactivated. And this is going to come to be important when we talk about this later on, because you're going to see that this principle is important in how some diseases manifest or don't manifest. Okay, but just right now, what to keep in mind is some genes you receive inactivated or imprinted. So maternally imprinted means they're inactive from the mother. Paternally imprinted means they're inactive from the father, okay? And so we saw the Punnett squares during Hiba's lecture. And so here, in this scenario, this is just a normal autosomal dominant Punnett square. So the father is affected, meaning he has only one affected allele, which is enough, okay? And the mother is normal, which is here shown by B. And so if you draw the Punnett square, you'll know that anyone with this type is affected, right? Because capital A in red here means that this person will carry the disease. So 50% of offspring will carry the disease, okay? And this we learned from, the basic lectures with patterns of inheritance, okay? So you're going to have 50% with the disease and 50% normal from this Punnett square, okay? And same thing goes here. If the mother is affected, you'll also have 50%, right? This B that's in red. And now you have something interesting when you have your epigenetic modifications. If you have the gene that you receive from the father that is repressed, where you have paternal imprinting, remember paternal means father, imprinting means you receive it inactivated, okay? Then even though in this case, you have your normal genes that are being received from the father, the mother has one single mutation. And so what happens here is even though you receive a normal gene here, because it is repressed or because it is not working, or I don't want to say not working, but not functional, let's say, the father's copy, it's as if this patient has the disease. Okay. So again, just to repeat, you have some specific diseases or some specific patterns of inheritance where you have maternal or paternal imprinting, meaning you receive a copy from the mother or the father that is inactive just at the get-go from the very beginning. And so here in the mother, you have one functional copy, okay, and one mutated copy. However, this specific gene or allele is paternally imprinted, meaning even before you receive it from the father, it's repressed. So when you do the Punnett square, you're going to have 50% of people that are normal, okay, and other 50% of people that are affected, okay? And this applies for paternal imprinting as well as maternal imprint. It's the same, okay? So here, if you have if the father's genetic phenotype is repressed, it's not going to matter, because even if you have a mutation here, this is not the copy that's functional. This is not the copy that's, let's say, working. So if you have that here, but this part is repressed, and you have that here, but this part is repressed, it is still going to be your normal copy from the mother that is going to be working. So that's why you have these specific patterns of inheritance that you see that might not be as traditional, let's say. But is this concept clear for now? This is another example. So let's say the other way around. If the father's copy is functional, okay, and the mother's copy is repressed, okay, and you have a mutation over here in the functional copy that comes from the father. But because you receive a repressed gene from the mother and a non-functional copy from the father, okay, you're still going to have an affected individual in this case and in this case. So 50% affected and 50% normal. Okay, does that make sense? And again, same vice versa. Clear? Or do you want me to repeat? All right. So this is what we call the parent of origin effect. So it makes a difference who you get the specific mutation from. For example, here, if you receive a mutation in something that is repressed in the mother, that means even if you receive this mutation, the dominant gene is still going to be the normal one from the other parent. So that's why it's important. You'll see some diseases and some pedigrees. That's what they're called. I remembered. And some pedigrees that specifically show, let's say, if you get this mutation from the mother, you get the disease. If you get this mutation from the father, you don't, and vice versa. And so that's why this happens, because some genes might be modified to be repressed, while the other ones would be functional, maternal and paternal imprinting. Okay, so that's what this might look like here. So you can see that here, this is a male patient that does carry the genetic component, and they pass it on to their offspring. Okay, however, in the females don't pass it on to their offspring. So the reason this could be happening is because this gene or this mutation is maternally imprinted, meaning the mother's copy is repressed. So it looks basically like this. All right. Wait, sorry. It would look more like, yes, like this. Sorry, if the father's copy is repressed and the mother's copy is mutated when you receive it, okay, you'll always be an affected individual, like here. Okay, so this goes the opposite way. So here, the males pass it on to their offspring, but the females don't pass it on to their offspring, but both females and males can be affected. That's how this differs from the previous ones that we saw. Okay, so you can see this again here. Males pass it on, males pass it on, males pass it on. And so this just goes generally. And you can see even here, females don't pass it on. Okay, here, even affected or non-affected females would not pass it on to their offspring. All right, and so the reason this happens is because of the concept that we're talking about, the parent of origin effect. Okay, so that basically the pedigree that we were looking at would look like this. Okay, because the mother's copy is repressed, there is maternal imprinting. Remember, maternal inactivation. So when you receive a mutated copy from the father, because this gene is repressed in the mother, you become an affected individual. That's why you're affected when you receive the copy from the father, okay? But you're not affected if you receive a copy from the mother because it's already repressed. So it's like the gene that is the problem is not being expressed anyway, so you're not going to manifest with this disease, okay? And so that's what we're looking at here in this example. And so again, this is what we call genomic imprinting. And some genes are expressed from the father, some genes are expressed from the mother, and some are silenced from the father, and some are silenced from the mother. Okay, and so different genes follow different patterns in that kind of way. And remember, and what's very important for you guys to know is that this is a normal process. Genomic imprinting is not only a result of disease. Okay, so it's a normal process caused by alterations in chromatin that occur in germline of one parent. So this is, for example, our maternal allele and our paternal allele. And some of the paternal parts are silenced, okay, some of the paternal genes, and some of the maternal genes are silenced, and that's why disease manifests differently. But remember, this is a normal process. This does apply to the way we look at disease and the way we analyze it, definitely. But just remember that genomic imprinting is normal, and they can definitely ask you about that in an MCQ. Okay, so just keep that in mind. And it is an epigenetic phenomenon. And so what epigenetic means, so epi is above, right? Like you guys took, what are you guys taking now? You're taking heme. Like, for example, epinurium, okay? And so above, and genetic referring to the genes. So it's something that does not happen with DNA mutations. It's an alteration that happens in the way you're wrapping your DNA around your histones and then forming chromatin. So how loose or tight it is around that. And this happens, and it's, again, a normal phenomenon, genomic imprinting. So that's what we call epigenetic. So it's a heritable trait. You can inherit it from your mother and father, but it is not the result of DNA mutations. Okay, and the reason this is important is because it has roles in a lot of different things within medicine. Intellectual disability, which was previously called mental retardation. It's also very important in cancer and why people are more susceptible versus others, as well as other conditions. And this is a very, very key field within research right now. And epigenetic modifications affect a lot of things, even why, for example, identical twins, why some may have specific personality traits while others don't, why some may inherit diseases while others don't. So again, it's a very important concept for you guys to understand. And I know we showed this pedigree a lot of different times, but that is one of the mechanisms potentially by which this could be happening. And then you have another very crucial concept. Is everything clear before we continue? Do you guys have any questions?
No, thank you.
Okay. And then you have something called uniparental disomy. So uniparental, uni meaning one, parental meaning parent, okay, and disomy meaning two pairs, okay? And so that basically explained the definition in a nutshell. Again, when you guys are stuck, just make sure to dissect the definitions in this way, okay? So what this means is you have a disomic cell line containing two chromosomes inherited only from one parent. So I'll repeat that again. So with these epigenetic modifications that we're talking about, what can sometimes happen is you receive two copies from your mother, okay? Or two copies from your father. Normally you need one from each person, right? And so when you get two copies from either parent, that's when uniparental disomy happens, and that can lead to manifestations of disease. The two key diseases I want you guys to know about are Angelman syndrome and Prader-Willi syndrome, and I'm going to talk about them now. So remember how we talked about imprinting? So what happens is normally you have your genes from your mom and your genes from your dad. And let's say that the genes from your mom are normally silenced, they're imprinted, right? You're going to get both, but they're silenced or they're not, let's say, functional, okay? And so when this happens normally, only one copy gets expressed. So only the genes from your dad would be expressed versus the genes from your mom. And remember, this is an epigenetic process. It's outside of the DNA. As you can see here, the sequence stays the same, but because of this thing called methylation or adding a methyl group, you essentially turn off this gene or you prevent it from being expressed. Okay. And so in something called Prader-Willi syndrome, you get maternal uniparental disomy. Okay. So if we saw here, we know the genes from the mom are silenced. Okay. And so if for some reason something happens and you receive two genes from your mom instead of one gene from your mom and one gene from your dad, well, we know the genes from your mom are silenced. So both of them, both maternally derived parts, are silenced. So that leads to the manifestation of disease called Prader-Willi syndrome. And this involves chromosome 15, by the way. I haven't seen that asked in previous questions, but for your knowledge, this involves chromosome 15. Okay, so if you get two copies of the silenced maternal gene, that's called maternal uniparental disomy. And what happens? In Prader-Willi syndrome, it causes intellectual disability. And when they're infants, they have very low muscle tone and problems with feeding. So they're basically very floppy babies. They can't really support themselves physically because of the low muscle tone. And when they grow up and become older, they have hyperphagia. So hyper meaning excessive, and phagia referring to eating. So they're excessively eating, and not like binge eating kind. It's they physically cannot stop. And so that leads to obesity, And they also have inadequate genital development and low IQ. And so this is a patient with Prader-Willi syndrome. And currently there are no specific treatments, but it does involve exercise and limiting their intake. But even in cases or families you see with children with Prader-Willi syndrome, they tell them, lock the fridge and lock the cabinets because they have no self-control. And that's because of this disease, because they receive 2 maternal copies instead of the paternal copy, which would normally be functional. And then you have the other spectrum. So on chromosome 15, you have 2 things. You have either Prader-Willi syndrome, if you receive the 2 maternal copies, or you can have Angelman syndrome. And so it's the same concept here. You have the paternal, which is normally silenced, okay? And you have the maternal, which is normally expressed, okay? But if anything happens to the maternal copy, if you lose it, for example, and receive 2 paternal copies instead, that is called paternal uniparental disomy. So it's the same thing we saw, okay? But instead of 2 maternal copies, you're going to receive 2 paternal copies, so 2 copies from the father. And these are both epigenetically marked, so they're going to be downregulated, let's say, okay? And so what happens in this case is something called Angelman syndrome. And it also causes severe intellectual disability, seizures and ataxia, meaning difficulty in walking, or their gait, or they're very unstable. And it also has unusual behavior that you can usually pick up on in a question. So they would say that this child is always happy, smiling, or laughing, even if there's no specific trigger or reason for them to be laughing. So they're considered very friendly, and that's why they called it Angelman. So just remember, these 2 involve chromosome 15. If you receive 2 copies, 2 paternal copies, paternal uniparental disomy, then you get Angelman syndrome, okay? And then in the case of Prader-Willi syndrome, you receive 2 maternal copies, all right? Is that clear for everyone? Okay. Do you want me to repeat anything before we go to questions? All right, I'm going to take that as a no. And so in this question, a girl has inherited both alleles of the same gene from her father. This is an example of essentially what we just talked about. So
unipersonal.
Exactly. So this is uniparental disomy. Would it be paternal or maternal uniparental disomy?
Paternal.
Paternal, perfect. All right. So basically that's what's happening here. So 2 copies of the same allele from the father. So this is paternal uniparental disomy. Follow-up question, if this was chromosome 15, would this lead to Prader-Willi or Angelman syndrome if there were 2 copies from the father? It would lead to?
Angelman, right?
Yes, Angelman.
Angelman.
Okay, the way I used to memorize it when I was in medical school is إنه الجنة تحت أقدام الأمهات. So like angels, and then you lose the maternal copy. It's a silly mnemonic, but that's just how I remembered it. Okay, so that's for this question. Now we're almost at the end, and then I have a lot of practice questions that are related specifically to this lecture for you guys. So now we're going to talk about mitochondrial DNA. So we know about your autosomes and your sex chromosomes, and also external to that, external to your nuclear DNA, you also have DNA in your mitochondria, and it's a circular molecule, okay? And it's very important for you guys to remember that it's 16 kilobases, okay? And that your mitochondrial DNA encodes for 37 genes. These are definitely things they can ask you guys in MCQs. So make sure to keep those 2 numbers in mind. So mitochondrial DNA is 16 kilobases, and it encodes for 37 genes, okay? And so the interesting thing about mitochondrial DNA is that you only get it from your mother. So essentially, the mother passes down her mitochondria, okay, in the process of fertilization. And as a result, if you have a disease in your mitochondria, you can only get it from your mother. You can't get it from your father. And this is an important thing. They might ask you an MCQ where they give you different points about the about human DNA in general, and they might say, oh, mitochondrial inheritance is from the father, and that's false. No, mitochondrial inheritance is only from the mother. So I used to remember it as like M&M, so you'd only get your mitochondria from your mom. Okay, and so because your mom can pass it down, it doesn't make a difference whether you're talking about females or males. It's going to be passed down to everybody, but only mothers can pass it down to their offspring. Okay, so this is an example of that. So you can see that, you remember, squares are males and circles are females. So males can never pass it down to their children because they're not giving them the mitochondria. But females can pass it down. You can see how here the mom passes it down to all her children, right? With no exceptions, because the only place you can get it is from your mom. Okay. And here females pass it down to their offspring. Okay, but males don't pass it down at all. So that's why there's a discrepancy here. Okay, and remember that all of your kids are getting your mitochondria. Okay, if you're a female. So all of them will have the disease. You can see here that there's not a single person that doesn't have the disease. So just keep in mind and make sure to know this Punnett square, this, I forgot the name again. I'll remember it in like 2 minutes. But remember this picture, remember the diagram very well, because it
Mitochondrial DNA.
Sorry, what is it called again?
Mitochondrial DNA.
Yeah, yes, the mitochondrial. No, I'm talking about the diagram itself.
Pedigree.
Pedigree. Thank you. Okay. Bye, let's go. You're gonna go? No. Okay. Yes. So this pedigree can definitely come in your exam. So make sure to know what it looks like, and just remember the pattern. You only get it from your mother, and mother passes it down to all her children. Okay. And so you have different concepts within mitochondrial inheritance, okay, called homoplasmy and heteroplasmy. So it's similar to when we think of penetrance. Okay, so homoplasmy means 100% of your mitochondria are mutated, or 100% of your mitochondrial DNA is mutated. And heteroplasmy, okay, remember hetero is different. And so here you have a bit of variation. And I'm going to show you guys this through pictures in the slides in just a second. But make sure to remember those 2 words because they're important in your exam. Okay, so this is your mitochondrial DNA. Okay, some of it is mutated and some of it is normal, right? It's not 100% mutated. If it was, we'd call that homoplasmy. Remember, homo, like homogeneous, everything is the same. Okay, and so what happens is you get clonal proliferation, and because of random segregation, some sections might have more of a mutation than other sections, and this is your normal phenotype. Okay, sometimes because of this segregation, you get even more of the diseased part. Okay, and this is the diseased phenotype. Okay, so basically this could be like a spectrum of disease. So the less blue you have, or the less mutated mitochondrial DNA you have, the less the disease will be expressed, or you'll have a normal phenotype. It wouldn't appear as though there would be any problem. But the more severe it becomes, or the more diseased or mutated DNA in your mitochondria you have, the more severe the clinical phenotype would be in this case. And remember that your mitochondria is everywhere. So when you have these diseased phenotypes, they can typically affect every system in your body. So it's not really limited to one thing or another. And so again, this is a concept that we call heteroplasmy. Okay, so just remember, if you have, let's say, 75% that's mutated, for example, that would manifest as disease. And then others, you would have less, and that's why you have less clinical manifestations. So that's why even though people might have the same mitochondrial disease, because of this different in percentage, you might have different clinical manifestations as a result of that. Okay, and so it follows basically what we already learned about in the very beginning: incomplete penetrance, variable expressivity, and pleiotropy. So remember, pleiotropy means it can affect various organ systems, which is what we just talked about, right? Because your mitochondria is everywhere. Variable expressivity means one specific symptom can be very mild in a patient or one specific finding and very severe in another patient within the same spectrum. And incomplete penetrance. So that means that maybe not everyone with a disease would get it necessarily. If you have, for example, 100 people with the same mutation in mitochondria, maybe 60 to 70% of them will clinically manifest and the remainder won't. And that's what we call incomplete penetrance. Okay. Any questions so far? Is it all clear? Do you guys want me to repeat anything? Because we got to the end of the lecture, by the way. So I hope it was clear and simple. All clear. Okay, alhamdulillah. All right, perfect. So now we're going to go through MCQs, and these may or may not be very similar to what you guys could get in your exam. And so first question is, which is a normal phenomenon? Okay, uniparental disomy is supposed to be A, but the options are uniparental disomy, widespread inactivation of the X chromosomes in a 46YX male, microdeletion in 7q11, uniparental disomy, genomic imprinting, or unstable triplet repeat expansion. So which of the genetic modifications you guys learned about is actually a normal process? Okay, yeah, you guys are right. The answer is D, genomic imprinting. So remember when we talked about Prader-Willi and Angelman syndromes, although genomic imprinting can lead to the manifestation of specific diseases when we talk about uniparental disomy, but the key thing to remember is that genomic imprinting, whether or not you have disease, is normal. Okay, so just keep that in mind. Remember, it was one of the things I kept in red for you guys, that it's a normal phenomenon. Okay. And this is just a pure recall question. The mitochondrial DNA genome is about how many kilobases?
16. E.
Yes, 16 kilobases. So remember 16 kilobases. And the next recall question you guys have is how many genes does the mitochondrial genome encode
37.
for? 37. Perfect. Okay, so these are the key things you guys need to remember. And anything in red is, again, stuff that has come previously. Okay. Which of the following is observed in mitochondrial inheritance? What do you guys think? Wait, let me fix something. 1 second. This really pissed me off. Announce. Okay. Sorry. Which of the following is observed in mitochondrial inheritance? I think you guys saw the answer when I exited the slide, but the answer is uniparental disomy. And why does this happen, or why is this the answer? It's basically saying the same thing that we know. You only get your mitochondrial DNA from your mother. Okay, you don't get anything from your father. And so that's, again, the concept of uniparental disomy. Okay, and just remember this. Exactly, only through the mother. So this just keep this in mind. Okay, and again, talking about mitochondrial DNA, this is another case for you guys. Khadija is a 33-year-old female who is affected with neuromuscular disease caused by a homoplasmic mutation in the mitochondrial DNA gene. So remember, she's a female, and this is mitochondrial DNA. The risk of transmitting the mutation to her son is almost 0, almost 100, 66, 25, or 50%. Okay, so remember, because she's a female and it's a mitochondrial DNA gene, remember the pedigree we looked at before, that means almost 100% of her children will get the disease. Okay, so if I asked you, what is the risk of transmitting the mutation to her daughter instead of her son, what would the answer be? What do you guys think? Still B, exactly. So your mitochondrial DNA does not differentiate male, female. It's not like your X chromosomes. Okay, so the risk would always be 100% of transmission because it is only female to male. But what if I flip the question and I wrote it this way? Tariq is a 33-year-old male who is affected with neuromuscular
Zero.
disease. Exactly, 0. And why would it be 0? Because males cannot transmit mitochondrial DNA. So no matter how many children he has, they will never get the disease because it's only from the mother. Okay, and so this is a very important question to keep in mind, and they really like to test about it. Okay, now this is a completely different question. We moved away. Okay, sorry, your net got stuck. So, okay, so if your mother is, if a female is transmitting the disease, mitochondrial disease, the risk is almost always 100%. Okay. Almost 100% because females can transmit it, okay? Only your mother is going to give you your mitochondrial DNA. But if I flip the question and told you it's a male who's asking what's the risk of transmission of a mitochondrial mutation, the answer is almost 0 because they're not transmitting to their offspring, okay? So I hope that cleared it up, okay. Why is it almost? Because there's a lot of things that can go wrong, and you know, I don't know the exact reason. I'll be honest with you, but in medicine we don't, in general, not just with this, we usually tend to say almost always, not 100%, not always, because there's always going to be exceptions. But I'll look into it and let you guys know if I do find anything about these specific cases. Yeah, there's no definite answer, but that's why it's almost 100%. Okay. So now we look at this question. You were consulted to evaluate a medical condition that affected several members of a large family. You observed that the inheritance of the condition did not follow classic Mendelian patterns. You also found that lifestyle and diet habits played an important role in the pathogenesis of disease. This condition is most likely sporadic, multifactorial, mitochondrial disorder, autosomal dominant, or infectious. So remember, multifactorial. There's genetic component that's important, but lifestyle and environment is also important. So this could be, for example, like the example I gave you guys about diabetes. It does not follow classic Mendelian patterns, but there is a genetic component. So lifestyle and diet does play a very important role. So if they ask you, this is how you should mention it. Or they could also ask you, what is the most common type of transmission? And you would say multifactorial. Okay, is usually the most common. Okay. Now, next question. You tested a sample from an individual with a mitochondrial disorder. You found that 76% of his mitochondrial DNA was mutated, while 24% was normal. This observation is called incomplete penetrance, pleiotropy, heteroplasmy, inactivation, or expressivity. What do you guys think?
A.
A?
Yeah, I think.
Okay. It's C. And first of all, I would like to just take this time to say thank you for making a mistake, because this is the best way you can learn. And I don't mean that sarcastically, I mean that genuinely. Okay.
Yeah.
Learning, okay, just taking this time to intervene to tell you guys, لا تخافوا إنكم تغلطوا. Don't worry about making mistakes, specifically when you're learning, because I'm not just saying this as Maryam, I'm saying this in general. It is scientifically proven. The more, if you get a question wrong, you're more likely to remember the answer than if you just get it right in the beginning. لا تخافوا إنكم تغلطوا. It's actually a very normal and important part of your learning process. You're not going to get everything right from the first time. Okay. So the answer is heteroplasmy. But let's go through every answer to understand why is this the correct choice. Okay, so you mentioned incomplete penetrance, and it would be incomplete penetrance if I told you that there are two people who are found to have the same mutation, okay? But one of them showed the disease and the other one didn't show the disease. That's when you talk about incomplete penetrance. When I talk about two separate individuals or a group of people with the same mutations, okay? So that's why this isn't the case here, because they're telling you this is one person. This is one person
Yeah,
order.
I got it. Yeah,
You got
okay.
it? So 76% of the DNA is mutated and 24% is normal. And so when there's a difference, that's what we call heteroplasmy. Okay, remember,
Yeah,
hetero means different. If it was
different, yes.
100% of his mitochondrial DNA, we call that homoplasmy.
Homoplasmy, yeah.
Okay, all right. And so the answer isn't pleiotropy, because remember, pleiotropy talks about when it manifests in different organ systems. Okay. Inactivation is when one copy is working and another is not. And expressivity is, remember, the spectrum within a specific clinical manifestation, like neurofibromatosis. Remember how some people can only have one spot and then other people can have, their entire body full of these skin lesions. Okay, I hope you guys aren't too tired. We still have some more questions. You examined several families with an X-linked recessive form of muscle disease. You found that most of the female carriers were normal, but some of them were mildly affected. This observation is most likely due to heteroplasmy, complete penetrance, skewed X-inactivation, homozygosity, or pleiotropic effect. So this is a bit of a harder question, but Hibba mentioned that you guys had a bit of trouble with this concept, so that's why I brought it up in this lecture anyway. But tell me, what do you guys think the answer is? Remember, there's always room for mistakes. I encourage it, if anything. Okay. C. All right, you guys are correct in your answer choice. And I'm going to explain why. Okay, so this is just what I want you guys to know. So females, we have two X chromosomes, right? And males, we have our X and we have our Y. And so just imagine in this case, the orange is abnormal and the blue is normal. Okay, so in an X-linked recessive disease, like the one in this question, okay, we know that females are more likely to be carriers, right? If males get one copy or the mutated copy, even though it's recessive, they immediately have the disease, right? Because they only have X and Y. So if they have the diseased X, khalas, they have the disease. But in females, it's a bit different. When they're carriers, okay, you have this concept called X-inactivation. So some genes on the X chromosome become stronger in one chromosome than another. So for example, let's put a pen, okay? So let's say we're talking about the gene here, and here it's mutated on the orange chromosome, okay? But even though I have the recessive copy of the gene, what happens is sometimes this copy gets inactivated, this chromosome gets inactivated. So the mutated chromosome isn't working, let's say, or is repressed. So what do I have? I have the normal version. So it's basically like I don't have the disease, right? That's why they have much milder clinical manifestations than others. However, if it goes the other way around, if for some reason my normal copy is inactivated, my normal chromosome, what's that going to do? It's going to be as if I have the disease, okay, even though I don't, because of this X inactivation. So it's as if my normal copy is repressed. So I manifest more of the diseased copy, right? And sometimes it happens the other way around. So if I have my recessive copy, okay, and then this part is inactivated, then I'll be more normal. Does that make sense for everyone? Or do you want me to repeat it? I can erase something on slide.
Can you repeat it again, please?
Yeah, I'll repeat it. لا تشيلهم. Okay, نخلي أحمر. طيب. If we're talking about X-linked recessive, okay, one, we know that it's more likely to happen in males. Why? Because they only have X and Y. So if they get one bad copy, khalas, they have the disease, right? Like imagine the orange is the mutated copy, okay? In females, it's a bit different because I might have my mutation here, right? And I have my normal and my abnormal. Imagine blue is normal and orange is you're mutated. So what happens normally is you have something called X-inactivation. It's as if one X chromosome is working or is stronger than the other. So in order for that to happen, one of your X chromosomes has to be a bit repressed. Okay, it has to, let's say, not be working as much, or this chromosome would be stronger. Okay, عشان تفهموها. So what would happen is, let's say I have, this is the recessive, right? And this is the normal. I'll just make it like a big R. I'm writing on like a computer. So what happens is, let's say my inactivation, my mutated copy is inactivated. My mutated copy is suppressed. It's not working as much. That's great for me because that means my good X chromosome is working, right? It's fine. Okay, so it's as if I have no disease. So that's why they're saying here, and you found that most of the female carriers were normal, right? But some of the carriers were mildly affected. This is why this happens. Okay, so let's say this is the scenario here, but let's say it goes the other way around. Okay, let's say for some reason my good X chromosome was inactivated. So I have less of my normal. And so what happens? I get milder manifestations of the disease. So even though I'm a carrier, I might clinically manifest the disease with mild symptoms. It's never going to be as bad as someone who has 2 mutated chromosomes because there's nothing normal. But basically, that's why they're saying in this question, some of the carriers were normal, but some of them were mildly affected. So this is because of skewed X inactivation. Okay, and it's just an important concept for you guys to know and a question they like to bring up. Okay, is that clear for everyone? Do you guys want me to repeat it again? Or all good?
No, I got it. Thank you.
Okay, perfect. We erase thing. Okay, so for now, I'm done. If you guys want to stay and practice some more, أهلا وسهلا, I have pedigrees for you to practice. If not, all of the explanations are يعني دايما موجودين. فعلى راحتكم. If you guys want to continue, we're more than happy to help. If you guys are capped out for the day, برضو عادي, no hard feelings. You guys will have access to all of them to practice on later. فقولوا لي whatever you guys want. حتى لو like شخص واحد طل في السيشن, I don't mind. أهم شيء إنكم تستفيدوا. You feel prepared for your exam. So let me know. What do you guys think? Can we try one? Okay. Let's try this one. Okay. So whenever you see any pedigree, I want you guys to keep in mind a couple points. 1, does it affect both male and female? Okay. 2, especially for the newer types of diseases that we learned about, is there a difference? Like, is it only males that can give you the disease or only females that can give you the disease? Because that's going to answer most of your questions, okay? And does it skip generations? Is, to help you understand, is it dominant or recessive, if that's the case? Okay, so now let's look here. What's one thing you notice? Okay, so this mother has the disease, or grandmother, I guess. Okay, she's going to give it to her offspring. Interesting. When the father has the disease, he doesn't transmit it to his offspring. Okay, but it affects both males and females. Interesting. Let's look at the other side.
The mitochondrial.
Exactly. When the mother has the disease here, or the female, she transmits it to all her children. صح? And here you see when the male has it, doesn't transmit it. Right?
Yes.
But the female transmits it to all her offspring. Okay, so only the female is affecting everyone, and everyone gets the disease. There's no exception. There's no child that doesn't have the disease. So that tells you that this is mitochondrial inheritance. Okay, so we said only females can pass it on, and both males and females can develop the disease. Okay, بما أنكم جلستوا, we'll keep going. But what about this one? we can see, okay, first of all, both males and females are affected, and they're affected equally. So we think, okay, maybe it's an autosomal thing, right? But males can pass it on, females can pass it on, but it doesn't affect everyone, okay? But it's present in every generation. For what do you guys think? Does this look like something dominant, recessive? How do you think about it? And again, feel free to make mistakes. This is your time to learn. Autosomal dominant, you are correct. Okay, so what I like about these little illustrations from osmosis is that they also write for you guys the genotype like TD or lowercase. So here it's autosomal dominant. How do we know that? First, like we said, it affects males and females equally. And two, it doesn't skip generations. And even when it's passed down with someone normal, here at like let's say 2/3, but a big percentage of the children are affected. Here, مثلا, half are affected. Okay, so an example of this would be Huntington's disease. Okay, and remember, Huntington's, for example, has 100% penetrance, so you don't need to worry about that. Mostly the pedigrees that they will get for you guys are straightforward. يعني ما حيجيبوا لكم penetrance and these things. It's a bit more advanced, but they might get you MCQs about it as a concept. Okay, so this is for autosomal dominant inheritance. Okay, now this one, and remember, 2 lines means consanguinity, so marriage within the family. Okay, so you can see someone affected here, right, in the great-grandparents, and then they don't pass it on to their kids. But when the family marries within itself, you start to get manifestations of the disease. Okay, and there's also males and females are both affected. Recessive, is it X-linked or is it autosomal recessive? Autosomal, okay.
Autosomal, yeah.
Because it's affecting your males and your females. And remember,
Females,
consanguinity
yes.
is very important to consider, especially in Saudi, because we have a lot of autosomal recessive diseases. Another way they could ask you guys a question about autosomal recessive is they could tell you, we want to start newborn testing or newborn screening on a disease. What is the, or what type of disease must it be? It must be an autosomal recessive disease for you to start screening. Cystic fibrosis, for example, sickle cell, thalassemias, these things, okay? Which we already have screening for, by the way, but just so you guys know as examples. So here you can see the affected great-grandparents, and the parents and the grandchildren are not affected, but they're carriers. And when you have a consanguineous marriage between 2 carriers, now you can have lowercase rr. Okay, and if they ask you, okay, you have marriage between 2 carriers of a disease, what is the percentage of likelihood that their offspring will get it? And it's always going to be 25%. Okay, so 25% or 1/4 are going to be carriers. 50% are going to be, 25% are going to have the disease. 50% are going to be carriers. And then the remaining 25% will be normal. اللي هم هدول. So disease 25, 50% carrier, and then another 25% completely normal. Both normal alleles. Okay, so this is autosomal recessive. Now this one. So we can see that you can have males and females affected. Okay. They can both pass it on, but you have a higher likelihood to have it in females. What do you guys think it could be? Autosomal dominant. Okay, any other guesses? X-linked. But X-linked, what? Do you think it would be dominant or would it be recessive? Because remember, even here, even if one person has the disease and the other doesn't, they're still likely to get it. Okay. So you're right. This is X-linked dominant. Okay, so you have members in every generation that are affected. So that's what tells you that it's most likely a dominant disease, first of all. Okay, and males, if you have a male affected, they will only pass it on to their daughters. That's very important to notice. Even here, you can see here there's a male. The dad will only pass it on to his daughters. He cannot pass it on to his sons. Why? Because they're going to give them the Y chromosome, not the mutated X chromosome. Okay, so all daughters are always affected. Okay, and the mother can pass it on to her daughters as well, or her sons. Okay, so this is X-linked dominant. Now we have this one. So, okay, interesting. It's not present in every generation, so not likely to be dominant. We can see it's only affecting males for some reason.
Y-linked?
Could be, but it could also be something else.
X-linked recessive.
Okay, X-linked recessive. But why is it X-linked recessive? So, first of all, it's, okay, here, this is the key thing. This is what's going to differentiate whether it's Y-linked or whether it's X-linked recessive. Here, if it was Y-linked, the father would always pass it to the son. But that's not the case here, صح? The son is completely normal. He doesn't have the disease.
Oh, right. I thought that guy is.
So here he passed it to his daughter, and the daughter is a carrier, which is why she's not affected. But she passed this copy onto her son. That's why he's affected. Okay, so mothers and daughters of an affected male are always going to be carriers. ليش طيب? Because this is the only thing that the father can give to the daughter. صح? He has to give her an X chromosome, and the other X chromosome is from the mother. So if you have a daughter and the father is affected, that means she's always going to be a carrier. Okay, so that's how X-linked recessive disorders present. How about this one? This is gonna help you differentiate between the two. So first, males are always affected, right? Males in every generation. Okay, and the father always passes it on to his son, but never passes it on to his daughters. So you guys are right. This is going to be Y-linked diseases, and so usually this is cases of baldness specifically, and I think also color blindness, but disease is present in every generation. Okay, and it's only passed on from the father to the son. Okay, in every generation again, because it's the only thing they have to give to the son. All right, this is just a summary slide of everything you guys need to know, because they're going to get you guys pedigrees and you're going to need to know how to read them. So I hope this cleared up everything. يعني من طرفي أنا وبرضه من طرف هبة. And if you guys have any questions, feel free to ask. We're more than happy to help. And yeah, so remember, dominant, you only need one copy to have the disease. Recessive, you need two. Okay, and in autosomal, it's equal in males and females. In linked, you can have X-linked or Y-linked. Y-linked, the father will always pass it to his sons, but never to his daughters. X-linked depends on whether it's recessive or dominant. But then you have mitochondrial. So remember, only the mother will pass it on to all of her children, and the father can never pass it on. And yeah, that's it. Any questions, you guys?
Can you send the slides?
Yeah, of course. I have, just so you guys know, except these are just extra slides I added at the end for the practice questions. But under all of the practice questions, you have explanations.
Yes.
And you also have the transcription of the lecture. This is the exact same lecture that Dr. Zahir gave you. I just added stuff. And remember, anything bolded and in red is very important. Make sure to memorize it.
Okay, thank you. Wallah, thank you so much.